Okay, so good afternoon, everyone. Okay, so today we will be start cestodes. Last few lectures we finish up nematodes, trichostrongyl nematodes, and important trematode parasites. So today and tomorrow's lectures we will be covered the important cestode parasites. So before we can start, I just want to see how many of you know the shape of cestode. Okay, so see, yes. So, cestode, we normally say tapeworms, phylum Platyhelminths, same like trematodes, but the class is Cestoda. So these are the most important veterinary and zoonotic cestode. So you don't need to be memorize all of these their class and families, but the most important cestodes like Taenia, Cainococcus, Anapocephala, Munisia, and Diphylidium will be covered that one in next two lectures. These are their final host, and those are their intermediate host. So the learning outcome, you will be describe the basic characteristics of the tapeworm biology, their life cycle, how these tapeworms has been diagnosed. You will be discuss the importance of these tapeworm treatment as well as prevention strategies in animals as well as in human. So drug of choice of in this group of parasite, and what is the significant zoonotic concern of these tapeworms. So these tapeworms are found in the small intestine in the case of definitive host. But in the case of trematode, we discussed that the definitive host will be these parasite, trematode parasite, found in the definitive host in the case of liver, in lungs, or intestine. But in the case of cestode, these cestodes are always in the small intestine of the definitive host. But in the intermediate host, that's the most common disease caused is in the intermediate host, and this is what we discussed in the next part of the lecture, what type of disease is caused by the intermediate host. But mostly asymptomatic disease is common in the case of definitive host. They are also hermaphrodite, dorsoventricularly flat. So they will not observe, they observe the nutrient because they don't have a gut. So the life cycle, they required intermediate host. Usually they have two intermediate hosts, sometimes one intermediate host, sometimes more than two intermediate hosts. So the body of this parasite consists of three main bigger structures. One is scolex, neck, and stubella. The scolex consists of a number of proglottides. These are the segment. So I'll show you in the next slide what these segment will be look like. Let's start with the scolex. Scolex is the head part, and that head part consists of a number of different structure like hook, that is here, and the suckers, they are here, and the neck is somewhere here. So these two type of adult worm are little different if you look it into the scolex. The scolex is like a slit shape here, but here is little bit different, and you can see hook and sucker in this type of cestode. We'll talk about more in detail why these two are different. On the other hand, stubella, the rest of the body part is called stubella, and each these small segments is the proglottide in Stobella. So is in some of the cestode there is a 2 to 3 centimeter long Stobella, and some has maybe very small 2 or 3 or 4 segments. So we'll also talk about which one is the small Stobella and which one has a longer Stobella. For example, here in the case of Tinea, we'll talk about Tinea later on. The Stobella is 2 centimeter long, is so big here, right? You can see all this is Stobella and consists of thousands of proglottides. So these small segments are the proglottides. In the case of Echinococcus here, you can see there is almost 6 millimeter long, is not bigger, and consists of 3 to 5 proglottides. So that's the difference between Tinea and Echinococcus. So you can easily differentiate at least these 2 genus based on their structure. So general morphology, what you will have to see in these Stobellas or in the segments, proglottide segments here, if that is an immature proglottide segment, you will see this genital organ here. It is just a starting point. But in the case of the mature proglottides, you will see more structure like testes, uterus. You will see the genital pores here, ovaries. So all the male and female sex organs are within the mature proglottides. So mature proglottides look like this, and immature will look like this. But in the case of the gravid proglottides, where you will see millions of eggs, they will call gravid proglottides. So you can see these small eggs are inside. So this is how these proglottides will look like. These segments will look like. So each individual segment will start producing egg at one time. Okay, so the eggs. There's a different type of eggs in cestodes. They are spherical, angular, or thick-walled eggs, and you always see in some of there is some difference between the eggs in the case of the eggs like Tinea type of eggs or Tineaid type of eggs, where you will see the oncospheres inside of the eggs. And here again, the small segments within the eggshell you will see the small segment of oncospheres. However, some of the cestodes groups, we will talk about that groups, you will see different type of egg. So that there is no oncospheres inside here. You will see the operculum type of egg. So here you have to remember that how you can differentiate between this type of cestode egg with the trematode egg, because they're very similar to trematode eggs. So this is very important point you have to remembered. So these proglottides are hermaphrodites, and one sex will be active at one time, and the cestodes reproduce a massive amount of eggs, but some of these will not be survived. Most of them will not be survived, but some of them will survive. And the larvae these are called metacestode. We'll talk about more in detail. They need to wait in the intermediate host. So larvae is always in the intermediate host, and this intermediate host is taken or eaten by the definitive host, and then this life cycle get completed. So these are the 2 groups of cestode will be discussed today and tomorrow. So cyclophilidine group or pseudophilidine group. Cyclophilidine groups, they have scolex containing heads, sucker, and hooks. This is what we have seen before, and you will see the proglottide segments in the Stobella, and one intermediate host. They always have one single intermediate host. Eggs, they always contain oncosphere. In the previous slide, we discussed about those who has the eggs who have oncospheres inside, and that belongs to cyclophilidine group. diverse larval stage. There's a different type of larval stage. We'll talk about these metacestode. We also called it metacestode. We'll talk about that one later on. Pseudophilidin group. So this is the one I have showed you. There's a slit or groove shape of head, more than one intermediate host, and the eggs are operculated always in the pseudophilidin group, and multiple larval stages within a single life cycle. Okay, so based on this group of cestode, cyclophilidin, pseudophilidin. So these are the number of different Taenia species in cyclophilidin group, Echinococcus species, Diphylidium, Munesia, and Anapocephala. So these we will cover those species in cyclophilidin in today's lecture, and then some of them we will cover tomorrow. And pseudophilidin group is Diphylobothrums, and Spirometra will be covered tomorrow. Again, life cycle. So that is the picture I have made by sketch to be understand the life cycle of this group of parasite. Instead of you can go into the Google it, look it into CDC or different. So it's more complicated pictures there, so difficult to understand. So because if you don't understand the life cycle, you will not able to understand the next step of the disease pathogenesis or clinical features and the treatment plans. So let's try to understand the life cycle of especially the Taenia and Echinococcus. Eggs are shed in the feces and they develop into oncosphere, and once they come out along with the feces, they will be start producing these. They have an oncosphere inside in the egg. So the intermediate host, let's say sheep in this case, they ingest this eggs containing oncosphere and further develop it into hexacanth. So this hexacanth then migrate into the predilection site in the intermediate host. Sometimes it's muscle, sometimes liver, or depends on different species, and then it will develop the metacestode. Metacestode is the larval stage here, and once ingested, this larval stage complete their development into adult once it is taken from intermediate host to the definitive host. For example, in this case, the dog eat the muscle, and they took this metacestode and it developed into the adult in the intestine. So this is how this life cycle works. So keep remember oncosphere, hexacanth, metacestode, adult stage. Right? Okay, so in the case of so this is the cyclophilidin life cycle, except in the case of Diphylidium and Munesia and Anapocephala. We'll talk about these species and the diseases these cestode cause. Arthropods are the intermediate host. In the case of Taenia and Echinococcus, always the ruminant will be the intermediate host. So the adult of the cyclophilidin group, as I said, the disease is developed asymptomatically in the definitive host. For example, Taenia, Echinococcus is the example here, and proglottides are rectangular or in shape, and we discussed about how this immature, mature, and gravid proglottides will be look like. In the case of the cyclophilidin egg type, what the egg will be look like, they always look like a Taeniid eggs. So remember, if you look it into the fecal flotation, you will not able to differentiate the egg of Echinococcus and Taenia. They are very similar. So you will be also says that this is a Taeniid type of eggs, right? And if they don't have oncosphere, cyclophilidin group like Taenia or Echinococcus, they don't have oncosphere. Pseudophilidin group always have a oncosphere egg, and they are completely different as compared to cyclophilidine eggs type. So here you will be in the cyclophilidine eggs. These are some of the example here, some of the eggs of different species. You'll see oncosphere here, but this is diphylidium. They have a small eggs within the bigger shell, and every egg has a small oncospheres, and this is a Munesia eggs, so that is a pyramid shape. Remember that these eggs are commonly not commonly found in the feces. You will be see these eggs. You will be seeing the feces. There is a number of white color ribbons type of structure, and that is the proglottides segments. That you will be see that segments in the feces. Once you see that segment in the feces, you will be take this segment out and squeeze this segment, and the egg will start releasing from that segments, and that is the much appropriate method to look it into the eggs or identify the eggs. In the fecal egg count, so you will be see a less amount of eggs. Sometimes you will not see the eggs. Interpretation will be goes wrong. So this is one of the best way to be look it into the eggs, to squeeze these proglottides, and in the petri dish put the water, squeeze it, they start releasing the egg from the segments. So this is the different type of cyclophilidine eggs. Either you will be seeing the flotation, or you will be do the squeezing of these proglottide segments. So Taenius and Echinococcus, they are single egg with striated shell, and oncospheres are in the middle here. In the case of Diphylidium, you will be see the egg pocket that contain a number of different eggs inside, and each eggs contain an oncosphere. In the case of Munesia and Episopola, they are always like D-shaped, triangular shaped egg, and piriform apparatus. So we talked about what the adult will be look like in the case of cyclophilidine group, what the egg will be look like. Now we'll be talk about the most important thing, that is the larval stages, what they look like, and we call them metacystodes. The larval stages in the tapeworms, we always use the word they are metacystodes. So there's a different type of metacystodes. Hydrated cyst. So it means that thick walled fluid filled bladder. So you will be see this fluid filled bladder, and that is a thick walled here, and multiple protoscolex is present within the cyst. So protoscolex. So this is the head part. Remember, the scolex is the head part. Once they are in the cyst, in the larval stage, they called it protoscolex. So you will be see this protoscolex, multiple protoscolex, and they are always, the metacystodes are always present in the intermediate host. And Echinococcus is one of the example of hydrated cyst. So cinereus, that is one other type of metacystode. Again, single fluid filled bladder, and multiple protoscolex will be present. These are the multiple protoscolex within the cyst, and they are found in the intermediate host. And sheep is one of the example. Ruminants are one of the example, and the Taenia multiceps is the only species they produce the metacystode called cinereus. In the case of Cysticercus, this is one other type of metacystode that has a fluid-filled sac like this, but there are only one protoscolex found in the cyst. So previously we talked about hydrated cyst and cinereus. There is a multiple protoscolex found inside, but in this case, only single protoscolex are found in the cyst. And sheep, cattle, pig, these are the intermediate host infected with this Cysticercus metacystode, and these are the number of different examples of the Taenia species. they produce this metacestode in the intermediate host. So number 4, cystosarcoids. This is also a fourth form of metacestode. Again, that is a single solid bladder fluid-filled sac and have one single protoscolex. So it means that hydrated cyst and cinereus is multiple protoscolex. In the case of cystocercus and cystosarcoids, one single protoscolex within the cyst. And they also find in the intermediate host, and that is the only cystosarcoid that is the intermediate host is the arthropods. So it means that Diphylidium, Anaphocephala, and Monesia, because these species, the intermediate host of these species are arthropods. Okay, so because metacestode are the important stage we have to be understand, because this stage is always in the intermediate host, except this cystosarcoid that is in the intermediate host is the arthropods, but rest of them intermediate host is the ruminants, and they cause a very severe disease in the case of ruminants, these metacestodes. So let's take out your point solution, and I really want to know how many of you understand this the concept of this larval stages. So which larval stage has a multiple protoscolex in the cyst? Oops, sorry. What has happened? No. I don't know, it's disappeared. Something happened. Anyway, okay, so cinereus and hydrated cyst is the multiple protoscolex in the cyst. So let's see this one. Which larval stage has a multiple protoscolex in the cyst? As a single protoscolex in the cyst. Okay, so cystocercus and cystocercoids. Great. Okay, so which of the following larval stages? Why? I didn't do anything. Oh yes, it's working now. Found in the arthropods. Okay, so cystocercoids. Okay, so up till now we finish up the important characteristics of the cyclophyllidine group, including adults, life cycle, adult, eggs, and larvae. Now we will be covered the basic life cycle that is in the pseudophylidine group. So in that case, dogs and cats, they are the definitive host, and they produce the eggs. And in the pseudophylidine, the eggs are operculated. They will not be look like the hooks the oncospheres hooks inside. So they will be more operculated and very similar to trematode eggs. These eggs will be taken by the first intermediate host, that is copepods, and the procercoids will be developed from these eggs. So these are the procercoids developed within the first intermediate host. So this first intermediate host then eaten by the second intermediate host, for example fish, and this procercoids will be converted into pleurocercoids. So this is the two main larval stages developed in a two different intermediate host in this case. And this second intermediate host, for example fish or rabbits, they are eaten by the definitive host and the life cycle get completed. So this is how the life cycle of the pseudophilidine look like. So a little bit different as compared to cyclophilidine, where you will be see a single intermediate host, mostly the ruminants, and some cases you will see arthropods, but here you will be see the copepods and fish are the intermediate host. So that's important. So now you can differentiate the eggs of the cyclophilidine group, that will be hooks or oncospheres, circular in shape. But in the case of pseudophilidine, the eggs will be more or less look like a operculum here and very similar to trematodes. So keep remember that. Okay, so what is the test, fecal diagnostic test we perform for trematodes? Sedimentation. What we will be going to do here? Is this sedimentation? No. Fecal float. However, one other best method other than fecal float, ribbon shaped proglottides will see in the feces. Take this one from there, put it into the petri dish with water, squeeze it with something or thumb or whatever you have. The egg will be start releasing in the water. Look it into the microscope and you will be see the eggs inside. Right? So that's the important point. So remember that no sedimentation test for these pseudophilidine group eggs. So the larval stage, two type of larval stage. In the cyclophilidine, 4 type of larval stage: Cineris, Cystocercus, cystocercoids, and hydrated cyst. Here 2 type: procercoids. Procercoids, they are found in the intermediate host, and intermediate hosts are copepods, and the larval hooks are, you will see the larval's hook are present. In the case of pleurocercoids, they are found in the second intermediate host like fish, snail, rodents. No larval hooks will be seen, and some stubellas will be seen here. Diphyllobothrum and Spiromatra, these are the 2 species that belongs to pseudophilidine group. Okay, so we covered 2 type of 2 groups of cestode: cyclophilidine, pseudophilidine. Now onward, we will be covered the most important disease that first belongs to cyclophilidine group, and tinea is the first one. That's really important. So these are the number of different tinea species: multiceps, hydatigena or ovis. Definitive host are dog. Intermediate host are sheep. Saginata and solium. Definitive host are human. Intermediate host are cattle and pigs. So let's start with tinea multiceps. So tinea multiceps are shed in the dog feces that further develop into oncosphere, and then this oncospheres containing eggs are ingested by the intermediate host, go into the blood to the brain of the sheep, and where the metacestode, or these are the larval stage, metacestode, larval stage. And remember, we talked about different type of larval stage. Here in the case of tinea multiceps, Cineris is the cyst formed in the brain of the sheep. And this cyst take 8 months to develop into within the sheep or the intermediate host, up to 5 centimeter in diameter and have a multiple protoscolex inside. So what is the clinical features? What this disease will be look like? We called it GID disease. So sheep will be show the circling. They will circle and head pressing. Their frontal region will be softened, and they have a wide stance and ataxia. So these are the number of different. Is a very nasty disease. Is very severe signs seen in the intermediate host. So look it into the intermediate. Dog are the definitive host, asymptomatic. Adult are there. In the case of intermediate host is cause a very severe disease, and lesion will be seen in the brain. Tinea hydatigena, eggs they shed by the dog, and they further develop into oncosphere, and this oncosphere ingest the intermediate host from via blood. First it goes into the liver, this oncosphere. From there, hexacanth develop, and this move toward the liver, or to further move toward the omentum, mesentery, and visceral peritoneum. So look it into all these organs. They are infected with the metacestode of this Tinea hydatigena. So this metacestode that will be there in the number of different organs, and they form a cystocercus cyst. So this is a single protoscolex inside in the case of cystocercus. Remember, we talked about that one. And this is one of the significant cause of the slaughterhouse rejection of a number of different organs, because it goes into the multiple organs where they form a metacestode. Tinea ovis. Again, dog shed the eggs, develop into oncosphere, and this is ingested by the intermediate host, and sheep is the intermediate host, and this time it goes via blood to muscles, and this cyst will be normally seen in the muscle, and then the oncospheres, they further convert it into hexacanth there, and then move from muscle to the myocardium and diaphragm. So it means that hexacanth is in the muscle, stage is the muscle, and then once they move toward the myocardium or heart or diaphragm, they further convert it into metacestode stage. And again, remember, metacestode is cystocercus here, is a single cyst in the heart and diaphragm. Again, common cause of slaughter rejection is very common. So onward, now we will cover the Tinea saginata that has a very important concern in the human, because human is the definitive host, produce millions of eggs, asymptomatic, but calves or cattle are infected with these eggs on the hand of these stock persons or those who are dealing with the cattle in the farm, and disease is normally associated with the poor hygienic condition in the farm. So the oncosphere, they go via blood into the muscles of the cattle, and then cystocercus bovis cyst is a single protoscolex cyst, that is cystocercus, that is developed within the muscles. And human become infected by eating of these raw meat from the cattle. Okay, so this picture we have taken from India. So what do you think? What is the risk factor of Tinea saginata in this picture? Anyone think about what is the risk factor here based on this picture? Just think about between human and cattle. Sorry? They're living together, right? So it's the same area, is a slum area of Bombay somewhere. They live together. So this is the risk factor there, right? Tinea solium, human, definitive host. and pigs are the intermediate host. They take this oncosphere containing egg, and poor hygienic conditions, that is one of the most important factor in this disease. Human become infected by eating of these raw or uncooked meat of these pigs, and this cysticercus single protoscolex inside can be developed in the human. They are accidentally ingestion of the egg. So it means that human can also act as a intermediate host in this particular Taenia solium case. Human is the definitive host, yes, but human is also act as an intermediate host by accidentally taken the egg from the feces from the human produce. The eggs, they can taken it and they can act as an intermediate host. So once they act as an intermediate host, think about how severe this disease will be, because intermediate host will be more affected as compared to definitive host. So here, what these risk factors are? Can you look it into this picture carefully? What you are seeing in this picture? Both. Anything? We are talking about pig. We are talking about human. What is the risk factor here? So see, these pigs, human, houses, pigs. So they are just living together. So these are the risk factors there. So once the human, they will be act as a, normally the humans eat the undercooked pork containing these larvae. They are act as a definitive host. The metacestode, they eat it and then they convert it into fine. But once the human can be infected with the eggs accidentally, then the cyst will be developed into the human brain. So that is called neurocystocercosis. That's a human disease once they act as an intermediate host. So normally once the humans serve as an intermediate host, the disease called neurocystocercosis is a very, very nasty disease, and normally seen in the number of different parts of the world like Mexico, Central America, Asia, and Africa. Some cases are seen in USA also. So this is the brain will be look like. So is a time bomb shaped brain. 50 million people has been affected. This disease is listed in the WHO neglected tropical diseases. More than 50,000 deaths per year. So this is a very, very important disease caused by Taenia solium in human. Here you can see the brain lesions. So diagnosis, you will be identify the eggs in the feces. Also, better option is to be, once you see these small proglottite segments here in the feces, that will be the best option to squeeze these one to take it out the eggs. And you will be say that one is a Taeniid type of egg. You can't able to differentiate the Taenia and Echinococcus without doing the PCRs. Praziquantel, epsiparental, these are the best drug of choice to control or treat the Taenia. No drug resistance issues in this case. So now will be covered the Echinococcus, one other group of cyclophyllidine group type of group. So Echinococcus granulosis and multilocularis, definitive hosts are dogs and cats. Intermediate host in the case of granulosis, cattle and sheep, and rodent in the case of multilocularis. Echinococcus granulosis, the definitive hosts are dog. They produce the eggs, and these eggs containing oncosphere, they are eaten by the ruminants or taken by the ruminants, and they will be survive as oncosphere will be survive up to 2 years. And you can see in the case of Echinococcus, you will be see multiple protoscolex and hydrated cyst will be there. So this oncosphere is ingested by the intermediate host, and then they move toward the lungs and liver, where the metacestode form is developed, and that metacestode form is a hydrated cyst in the liver, lungs or the liver. So this cyst will be always found in the meat inspection, and the distribution of echinococcus is normally in the USA, in the Arizona, New Mexico, northwest part of the USA. So these are the number of different area you will be see the distribution of the echinococcus in United States. Echinococcus multilocularis, definitive host are dogs and cats and sometimes wild canids. Intermediate host are rodents. However, large ruminant like cattle and human can also be a intermediate host. They can also be infected from the eggs from the feces of dogs. So what happened in this case? The intermediate hosts are infected by the oncosphere containing egg, and this larvae develop within the liver. So this is the very, very big hydrated cyst normally seen in the liver. This is gelatin matrix seen in where you will be see a protoscolex, number of protoscolex within the hydrated cyst. So look it into this pathology. So these are the cysts, how big they are, right, in the liver. So this is really, really nasty disease once the cattle and sometimes human has been infected. But rodent are the most important intermediate host. Distribution, you will see that one in the northern Hampshire, like Canada. Normally this will be spread by the relocation of the foxes and the also higher prevalence in the Arctic region. Okay, so now let's do some knowledge check questions here. So select the definitive and intermediate host of Echinococcus multilocularis. Okay, so dogs and cats are the definitive host, rodent and sometimes cattle and human are the intermediate host. So in the case of the in the H-wise, normally this is Echinococcus granulosus. Diagnosis, you will be see these tinea eggs, very similar, tinea type of egg, very similar to tinea. This Echinococcus is very similar to tinea. So we called it teneid type of egg. So must treat when you will find the teneid type eggs. Probably they are tinea or could be Echinococcus. So praziquantel, these are the drug of choice in this case, and there is no any resistance against these drugs. This is one of the best drugs you can use for the control of Echinococcus infection, either multilocularis or granulosus. Okay, so anyone has any question? Because this the life cycle, the difference between cyclophyllidine larval stages and pseudophyllidine larval stages, these are really, really important. Without knowing these stages, it is difficult to be understand the clinical features or the pathogenesis of this disease or the treatment plan. So these larval stages are very, very important to be remembered. So based on this, you can further look it into the clinical picture of these diseases. So anyone has any comments or any question? No? All good? So just to be remind you, we have review session that's supposed to be Friday afternoon here. Okay, great. Thank you.